Having seen the anatomy of eye, now next we are going to deal with the mechanism of vision. As you know, the image is going to be formed on the retina. So first of all, let's talk about the cells of retina, how the cells are arranged in retina. If you have to dissect a retina, this is my way of presenting retina. If you can understand it, little bit distorted again it is, but what you see over here is that there are three layers of cells. Cells are arranged in three different layers. First layer is those of photoreceptor cells. Okay. The second is of bipolar neurons. As we started with the chapter, we had discussed that bipolar neurons, or if you can recall, we did discuss about types of neurons. One of it was bipolar, and we said it that it is present in our eyes retina. We have those bipolar neurons over here. We have ganglionic cells, and this is the optic nerve that is being formed, which would go to the blind spot. Now what you see over here is this entire is retina if it is magnified. So supposedly this entire huge thing is the eyeball. Try to make it out. This is the pupil. From here the light comes, it comes over here, it strikes these photoreceptor cells, it has to reach this part, then this part will take it to the brain where the image would be processed. Now you see this entire whiteboard can be considered as the human eye, and this is the last layer, that is retina. It would be followed by choroid and sclera in other terms, but we are seeing this retina highly magnified at this stage. Now what we are going to see is that how image would be formed. For that we are going to completely understand the various layer of cells of retina, because ultimately this is the screen where the image would be formed. So let us see. These are the rods. Photoreceptor cells are present in the retina. The first layer is of photoreceptor cells. There are two types of photoreceptors. One is rods and other is cones. Okay. So these rods and cones are the photoreceptor cells which have photopigments in them. Different types of photopigments that would be present in these photoreceptor cells. Now the first one being rods, you have to remember that different types of cells are responsible for providing different types of visions to the eye. Rods are responsible for scotopic vision. Now what is this scotopic vision? This scotopic vision is the vision that you get when supposedly you go out in dark. There is a little bit of light that is present. The vision that you get in twilight, you are able to distinguish the things, but you cannot make out the color. You are getting my point. Supposedly you go out in the evening when it is twilight and you see the things, they appear dark to you. You are seeing a tree, it would appear dark to you, but you will not be able to make out what is the color of the leaves. But given that condition, you are asked to see the tree in the daylight, broad daylight, you will be able to make out the color difference in that tree, that it is having green leaves. So that is that vision where you can make out the color difference. That vision is photopic vision. Okay, and the one where you cannot make out the color difference, the thing is just more dark, light, lesser dark and somewhat light, that is known as scotopic vision and photopic vision. Now I hope you can make out the difference between the two types of visions. What you have to learn over here is that rods are responsible for scotopic vision, where the point of difference between the images formed is in terms of the contrast, whether one is more dark, less dark, or lesser dark in the much lesser dark, or the darkest. In that way you have the scotopic vision and rods are responsible for that. And in photopic vision, the cones, photoreceptor cells called cones, are responsible for the photopic vision, where there is the image that is formed that will be giving a color identification, and cones are responsible for that. You can learn it in terms of C is the first alphabet for cones and C is the one for colored as well. So we have a colored vision, like in the case of broad daylight, you can make out the difference in colors. Now, when we talk about rods, we are talking about scotopic vision. Have you ever heard about night blindness? You study about it from your junior classes, that night blindness is caused by deficiency of vitamin A. If you haven't studied, at least I studied it. I learnt that night blindness is something from one thing, that vitamin A's deficiency is the causative factor for that night blindness. Night blindness is caused when we do not have vitamin A in our diet. And why do we need vitamin A in our diet? The reason is that vitamin A's derivative known as rhodopsin. Rhodopsin is a pigment that is present in the rods responsible for scotopic vision. Now, if somebody is having night blindness, it will be due to deficiency of vitamin A. They will not have rhodopsin in their eyes and it will be difficult for them to see during dark. Now, coming to the cones. These cones are giving you colored vision. You can make out the color difference. This is reddish, this is green and this is black, as you can see over here. Had there been no light or dim light, you will not be able to make out the difference. It would appear a dark color to you. Why? Because cones are responsible for this particular cones have 3 different types of pigment cells present in them. The different types of pigment cells are responsible for perceiving the light in 3 different colors, that is red, green and blue. In case all the cells are stimulated at an equal level, then what you see is white light. So these cones are for resolution, a well resolution image, and we did discuss that what is the point where maximum resolution of image could be achieved. That was fovea centralis in macula lutea, where maximum cones were present. More would be the number of cones, more would be the perception of light and more would be the more clearer would be the image. Now coming back to the mechanism of vision. See, this is all the introductory part. Now what is going to be in mechanism of vision? These cells are to be stimulated. The stimulation is going to generate some sort of impulse. That impulse has to go to the brain through optic nerve. So that is what has to take place. Now you know these rods and cones, one thing is missing over here, that we have at times amacrine cells over here and horizontal cells. See, amacrine cells could be present for passing the information here and there, which would be in connection, and horizontal cells would be present towards this part. Again, these horizontal cells would be developing some sort of connection, just for making you remember the names, that they are present in such an arrangement which are maintaining a connection between all these cells, amacrine and horizontal cells, and you have to remember that these are present inside the retina. These 2 names are present inside the retina. Now coming to the mechanism of vision, it is going to generate some sort of nerve impulse. Now you know how a nerve impulse is being generated. It is generated when a potential difference is created. Polarization is being created. That has to be done by a potential difference generation. The potential difference would be created when the light comes and falls on these cells. You know, these cells, they are having a pigment known as opsin, or its derivatives, as we had rhodopsin and retinal. These are the 2 pigments which are present. Now this opsin is a protein while retinal is an aldehyde. Biochemistry, talking about their biochemistry, we have these 2 points, that opsin and retinal. When the light comes and strikes on the rods or the cones, the opsin gets dissociated from the retinal. They both are present together. The opsin would be dissociated from the retinal by the stimulation that has come by the falling light on the retina. As soon as the opsin gets dissociated from the retinal, there is change in the structure of opsin. When there is change in the structure of opsin, what happens is that an impulse is generated and the bipolar neurons are ready to take that impulse. Now the nerve impulse would be starting, it would go to the optic nerve, it would go to the optic chiasma. Over there are optic parts of the brain which perceive the image. The image which was made over here by the photoreceptor cells, inverted image as it was, it would be processed by the brain. We are going to see the erect image, and how that image is getting the colors and how we are able to resolute it, that is under the mechanism of vision, where the light comes and falls on the photoreceptor cells. Photoreceptor cells have a photopigment, it dissociates, it generates an impulse, and that impulse is further sent to the brain for further processing. So this is all about mechanism of vision. It is quite a complex mechanism, but what we restrict ourselves to is the understanding from the point of view what type of cells of retina are playing what role.